Hello, my name is Nathan Rayna and I'm at Ouachita Baptist University. Thank you so much for joining us on our first virtual AR Cure Workshop, or Arkansas Cure Workshop. I need to thank the people who helped me put all this together. One is Dr. Ruth Plymell. She's been my great friend and colleague for nearly 10 years. She helps me organize, put all this together. She helps me develop cures. We develop everything together. And I cannot do this without her help and support. I also have Dr. Jamie Sable now from the University of Memphis. She's a science education specialist, and she's helping with assessment and putting all these things together and assessing, are we actually learning anything and accomplishing any goals. AR Cure is funded through an EPSCoR grant, which is through NSF, the National Science Foundation. And the particular EPSCoR grant funds called the Center for Advanced Surface Engineering. And so I definitely have to thank my funders and benefactors for helping us put together this workshop. AR Cure has had 3 different workshops so far. So we started in 2017. In 2019, we had our biggest workshop. We had over 30 faculty. And we've had 62 total faculty have attended the AR Cure workshop over the last 3 years. And it began as part of Arkansas EPSCoR. And so we needed a way to increase the broader impacts of this grant. And I brought it up to them was, why not take what we're doing in the classroom and create a workshop around how we would do these types of things. And we also was an outbuilding of Malcolm Campbell's Genome Consortium for Active Teaching. He had a synthetic biology workshop for many years. He let me use his format and his ideas, and we took what he's created and we've incorporated it into what is now the Arkansas Cure Project. It all began when we're at Ouachita, when we wanted to try to increase our research. We really wanted to figure out how to get students more involved with research. And so we wanted to encourage more students to participate. We wanted to improve the quality of our research. We have, for a while there, I was doing these things that felt very science fairish. And how do I get something when I take a kid to a meeting, they're going to stand out? And really the main thing that we were trying to have is, how do we make our kids more competitive? We want our kids to be competitive for off-campus opportunities. When they go someplace, we're from Southwest Arkansas. I want other schools to see what they do and go, wow, this kid is going somewhere and I want to work with this kid. And so we really had this large focus of what we were trying to do, all centered around research experiences. And so right about the same time, the Vision and Change came out and sort of called for the same type of ideas, where students need to be thinking, they need to be creative, they need to be expressive, they need to be thinking like a scientist. The whole goal of everything we do in AR Cure and all the cures that we work on is having students think like a scientist. That's the #1 goal that we want to look for. And when we go to design our Cure lab, we really move from this cookbook lab, right? I used to do this lab and they would watch the things and the plants would get big or something would turn green. And we always call that sort of a cookbook lab. And those things have their place and they have their benefits. And so I don't necessarily want to degrade them, but we wanted to move from that into what we think of as a real discovery lab. And we want to take all the different components that we see in an independent one-on-one research experience, and how do we scale that into a large classroom where we have 40 students all interacting on different projects. And so that becomes the foundation of what we do when we create our cures. And when we think about cures and we think about what we've done, Ruth and I sat down and started making these ideas of what makes a good cure, right? What have we observed over the times? And others have published these and very similar ideas over the years, but we feel like a cure needs to be very unique. We want to create a sense of ownership. We want students to feel like they are doing something new and novel that no one else is doing, that no one other group is doing. We want that to be really owned by our students. We want to have a sense of community. Science is a community. And so we want our students to not only feel like a community within the classroom, but we want them to realize that they're part of a larger community within the United States or even the world. We also need to have a manageable number of techniques. We're not getting caught up in all the different techniques that we think a student should observe in a genetics lab. We're not worrying about those. We're concentrating on thinking like a scientist and we want a manageable number of techniques. However, we also want this to be challenging to our students. And so we don't want it to be easy. They can't get done in 1 week. They have to have problems. They have to have multiple iterations of these techniques. And we realize that we think students need consistent project goals throughout the semester. I know a lot of people do modular-based cure labs, and I have no problem with that. And every school and every situation is very different. But in our hands, in what we do, a very consistent goal where the students are working on a project with many sub-projects to get to a final goal is what we think works the best. It also, in my hands, makes a much better lab notebook, and it makes it much better presentations that we'll see later on. At Ouachita, we have scaffolded cures across the entire curriculum. We have cures as freshmen. We're part of the HHMI CPHAGES program. We were cohort 4, so we've been there for a while. Ruth has also created her own cures that we now are embedding with all of our freshman biology majors. Ruth is involved with the microbiology, and she's part of the Small or Tiny Earth project. We have a cell biology cure, which is part of the Cell Biology Education Consortium, or CBEC, and we use cell blocks to create different research models where students can use tissue culture, and they do cancer tissue culture in my cell biology lab. And I can tell you more about CBEC later. And then genetics is our synthetic biology cure. Students do all these different cures. Not every student takes every one of these classes, but most students take at least 2 of these cures throughout the course of their career at Ouachita. And from there, they all take a senior research capstone class where they have to do an independent research project. A lot of times these projects are their cure labs been expanded into something they want to work on further as they move forward. At Ouachita, in the genetics is a senior level class, really junior senior level when you start looking at the hours and who takes it. Approximately 40 students I have each spring in my class, and it's a big class. We don't use multiple choice. We have a lot of sort of essay and critical thinking within the classroom, but the lab is where the synthetic biology is incorporated. I have 2 labs, so if I have 40 students in a class, I'll have 2 labs of 20. It meets for 3 hours a week. And what students do is in the lab is they work as mini iGEM teams. They work as groups of 2 to 3 students. I find that 2 is a good number. 3 is not bad. 3 helps to reduce resources and still keeps quite a bit of one-on-one work going on. And what they're doing is mirroring or managing what an iGEM team would do. And so iGEM stands for International Genetically Engineered Machine, and it's a competition at MIT. And for the last 15 years, this thing has just grown and grown and grown. Last year, I think they had over 300 teams. Each team had between 8 and 10 students show up to this giant jamboree. And each team is working on some sort of synthetic biology component. They're trying to build new devices to measure something in nature. They're trying to make new devices to make drugs. They're trying to do all kinds of different things within the synthetic biology context. Well, they may do it over a course of an entire summer with a large group of people. Our students are doing it in groups of 2 or 3 on a much more smaller scale, but we follow the exact same model that iGEM puts out for their competition. And so the way to think about this is we keep talking about synthetic biology, and we'll get into all the different aspects of synthetic biology. But many of you are probably aware with cloning, and you've cloned parts together. It took me an entire year to put my first constructs together when I was making transgenic plants in graduate school. However, these parts have been standardized, and so they're real easy to put together. And we think about the assembly line as being the big thing for Henry Ford, but really what made the assembly line possible was his idea to standardize all the nuts and bolts and parts. And now people, no matter where you're at in the assembly line, you could grab a part or nut or a widget from a different part of the assembly line and it would work for whatever part you were building. And that's exactly what's going on with these standardized parts. And so we're using all the different promoters and ribosome binding sequences and reporters like green fluorescent protein, but they've been standardized where we can interchange them and put them all together in a very uniform fashion. This is easy for students to understand once we teach them the process. And so The way I always talk to students about this is when we think about going to something like Lowe's or Walmart, right? Or you go to Lowe's and they got a row of nothing but light switches. And I love the place. We're remodeling my house and they're just light switches. And I pick which one I want. Do I want a round white switch? Do I want a dimmer switch? Do I want a toggle switch? And don't tell them where those things came from. That's just like a promoter. There's all kinds of promoters that we can work with. They come from all kinds of different organisms and all kinds of different areas. But they've all been taken into this one form and then turned into a standardized biobrick, which then can interact with other biobricks. And that's exactly what Lowe's or Home Depot does when it sells all these different parts. They've all been standardized, and now you can mix and match them. And now I can have dimmer switches in one room, and I can have motion sensor switches in a completely different room, and they all interact with each other. Now, something to think about is degrees of complexity. People always ask, well, how complex are your projects? And if you go to the IGM websites, those things are really complex, and they're amazing. And I think about building Legos with my kids, and we'll get these kits, and they'll have these giant, awesome cars that we could build. But we never build those cars. We always build some essence of those cars. And that's really what we're doing in the undergraduate classroom. We're building the essence of what some of these larger IGM teams are building. But remember, these teams are doing it in groups of 8 to 13. They have 2 or 3 faculty advisors, and they do it over a course of a summer, if not an entire year. We're doing this with students working in 2s or 3s over a 10 to 14 week period. And so sort of how we put all this together is we have the students go online and start investigating synthetic biology. There is a lot of material on the IGM website. Sense of community. They go, they see what's going on, they think about how all these things are working, and that is huge for them to understand that they are part of a community. They also learn to use the IGM Registry of Standardized Parts. And so this registry is amazing as far as the amount of information that it has on it. As people use the parts, they can go back and post more information or links to where you can see their data from those parts. And students design their devices. We have multiple presentations at this point. Students are always tasked with standing up, presenting, and showing something related to IGM and related to how they would design their parts. Once again, communication is key as being a scientist. I also show you how to join the IGM Labs program. The IGM Labs program is where you can join for $500 and you will get all of their standardized parts. You get about 3,000 individual plasmids. I let my students mix and match these plasmids as they see fit. And we'll talk about funding and how we get those things put together later. We do have a large wet lab component, have an online lab manual where we've put together, we use Zymo Research's protocols almost exclusively. However, we've gone back through and we've made notes for the students where essentially at this point, you can put it in a freezer and wait till next week. If you start at this point, you can't stop. You have to keep going. And this allows students to start planning their day. It is important before they come to the lab that they plan their day. Back when I was doing these sort of cookbook type of labs, they would show up, they weren't prepared. I would help them do it. It'd get done in 2 hours anyway, and we'd roll on. Here, they have to show up unprepared. If they don't show up unprepared, they don't complete the lab. I don't care if they don't, what happens that day. Because the grading is on a benchmark process, and they just have to complete the project. Some students take weeks to do it. Some students complete it, never complete it. And so it kind of depends. And they have to self-regulate and realize how to manage their time. The other deal is that since our techniques are relatively simple and relatively manageable number, they will do these techniques multiple times. 10, 15, 30 times is not unheard of. But if they have a problem, they have to stop, they have to think about what they need to do, and they'll go back and fix that problem. And that, once again, is thinking like a scientist. They also have to analyze their device. They have to design some sort of experiment to measure it, and they have to research that as well. And so if you want to look at a project overview of how this happens over the course of a roughly 13-week semester, is they begin working in the researching the field. They begin working online. The first 2 weeks is almost exclusively online with multiple presentations. We then start introducing the online lab manual. where they learn the techniques, and about 3 to 4 weeks into the semester, I'm turning them loose. They're now have learned all the techniques. We can learn more as they get to them. So I don't teach every one of them the same technique, but they sort of, oh, it's time to run a gel. Come over here, I'll show you how to run a gel real quickly. You can watch the videos on how to run a gel, and then you're on your own. And then you can teach somebody else, right? So now we're being more of a sense of community. We still want to create a functional device. A functional device, the most simple functional device, is a promoter, ribosome binding sequence, and some sort of protein, usually green fluorescent protein, red fluorescent protein, blue chromoprotein. But once they create that, they then have to analyze that device. So now they have to go back, they have to design experiments, and they have to analyze it. Some of these experiments, they use my plate reader, and they design really amazing types of projects where they're using a plate reader, measuring fluorescence at different times. But I've had other students who didn't want to use a plate reader and essentially set up a scale system to look at fluorescence, and had other students in a hallway do 1 to 5. Is this one a brighter colony, or is this one a brighter colony? They designed a perfect experiment, ran statistics on it, and it all worked great for them. So here's just a project example that I shamelessly stole from one of my kids. This is some parts that were through iGEM. And so what we see here is we have the Lac Promoter run by a ribosome binding sequence with green fluorescent protein. And we have 2 terminators here to separate it from an arabinose inducible promoter, ribosome binding sequence. And then what we can do is clone in different Lac inhibitors. And so this is a pretty common device that you can find on the iGEM website. However, we stepped it up a little bit because now we add some bioinformatics where we take the variables of the Lac inhibitors, we look at the sequence, and students can actually go through and change these sequences. And for about $50 to $60, we can have these parts actually made through IDT DNA. And within a week, we get the parts, students clone them in. And what's really cool is when they clone these parts in, we can add a arabinose, we can induce our Lac inhibitor, and if it's produced and it's functional, it'll go back and it'll bind to our Lac promoter, and it'll prevent GFP from being produced. So now students have a way to measure green fluorescence here. They have a way to activate their Lac inhibitor, and now we can actually measure how well the sequence that they designed, that they put together, is actually making a protein that's interacting with our promoter. Now there's all kinds of different learning outcomes that you can start discussing here, right? We're talking about promoters, we talk about gene expression and class, and we're always bringing up class examples back into the lab. And in class, we're always talking about what we're doing in the lab and bringing those things back and forth. And so there is a lot of different versions that you can create. And so my students over the last few years have made multiple versions of this where we can control the different Lac promoter, different versions we control the PBAD promoter. It has its own regulators. Students this semester, we went online, found a ton of different promoters and of all these different variables that we can start designing and creating new projects. And so this is just an example of a project that was completed in a semester. A lot of the parts were already built on the iGEM website. And so a lot of it was going and finding the pieces that you want, putting them together in an order, and then testing it. And I do have to admit, later on, we'll talk about problems, and more problems. I probably can tell you more things not to do than I can tell you things that can do. I've been doing this for since 2009, and I have slowly gotten better over the years. And then the next year, I feel like I forgot everything I learned. And so we are definitely not experts at this, but we can tell you what not to do. And that's really one of the things that we'll talk about as we go through this. We'll also talk about how to get student and faculty buy-in. The first few years we did this, buy-in was not very high. However, as we start getting some notoriety, as students are getting into programs, as a result of doing these labs, we would encourage them to put them on their transcripts and their resumes, and now they're really starting to buy in quite nicely. And so that concludes our first meeting of the AR Cure workshop. I once again have to thank all of my supporters. I need to thank Malcolm Campbell and the Genome Consortium for active teaching. It's his project that we're really sort of built out as the AR Cure, and so I really appreciate him for all his work and effort to allow us to do this over the last 3 or 4 years. We'll see everybody later. Thank you.